In today's video we are looking at DNA structure and function, and this is for the higher tier for GCSE Biology Unit 2, which is sometimes used for additional science as well. Now, in order for us to be able to understand the structure and the function of DNA, we need to be able to look very closely at the DNA molecule itself. So what we're going to do is we're going to start off quite large. We're going to start off at the cell, and this is a cell from a human. We'll pull out one chromosome and magnify it. So it might look a little something like that. If the cell is actively dividing, it might be doubled up, but for now I'm drawing the chromosome like this. Now, for us to understand DNA structure, we need to magnify a little bit. So this blue section here is part of that chromosome that's been magnified, and you'll see it looks like a long piece of string that's been wound up or coiled up. In actual fact, that's what happens. The string here, the purple bit, is the DNA, and it has been supercoiled in order to compact it into a chromosome. But that's not far enough. We still need to magnify a little bit more. So I've unraveled the DNA here, so it looks like a piece of string. But if we then magnify further still, you will notice that the DNA is actually two strands that are wound around each other, and this structure is called the double helix. So two strands wound around each other, but you'll also notice there's these parts that are protruding or poking inwards. So if we unravel those and have a look at those a bit more clearly, what you'll see is those two strands that we've been talking about. We've untwisted the DNA molecule so you can see it a bit more clearly, and what we're going to look at is, or what we're going to focus in on a little bit more, is what's going on at the ends of these parts that are protruding inwards. So here is the section of DNA here. I've just got this diagram just to remind us what sort of level we're looking at. But here's a part of the DNA molecule, and I'm representing this as a gene. A gene would be a short section or a small section of the DNA. And its actual job, its actual function is that, short section of DNA that codes for a protein. In actual fact, it might be a whole protein, it might be part of a protein, but the point is that the genes in the DNA, what they do is they carry the code to build proteins, and we're going to look at how they do that now. Now, these parts that I was referring to earlier are actually called bases, and there are 4 different bases. Their names are adenine, cytosine, thymine, and guanine. And while you're probably not required to remember or memorize these names, it's probably worth being aware of them. So if you do see them in a exam paper or in a book, you actually know what they're referring to. But usually we refer to these just by the letter. So A will be adenine, C is cytosine, T is thymine, and G is guanine. And what we will find is along the section of the DNA, along the strand, we'll see these bases that are in a particular order. So this might be C, T, A, T, A, C. So the order might look a little something like that. Now, on the other side, on the other strand, we also find the bases, but we find what we call complementary bases, and that means whatever base is on this side must complement the one that's on the other side, and that basically means a C can only pair with a G, T can only pair with an A, and therefore opposite this A here would be a T, opposite this T will be an A T there, G, C, T and G. Okay, so these are the complementary base pairs, but when we're making our proteins that we've been talking about, we only look at one strand. Only one strand of the DNA is used. Now, the way this works is we have what we call triplets of bases, or 3 bases at a time, and each triplet, or each of the 3 bases, codes for one particular type of what we call amino acid. So these are triplets, and this triplet here, CTA, will code for one particular type of amino acid. And as we know, or you should know from previous videos, amino acids make proteins. These are the building blocks of proteins. So if we know what order to put the amino acids in, we can build a particular protein, and there are thousands of different types of protein in the body, and if we get the code, we can have lots of different codes that will build all these different amino acids. So just as an example, CTA codes for amino acid 1, TAC will code for amino acid 2, and what the cell actually does is read off these codes and puts the amino acids in the order that the code said in order to build the particular protein that's required. So here I've done a slightly longer version. Here's a slightly longer gene, but this code here, CTA, TAC, GAC and so on, is instructing the cell to put the amino acids together in this particular order. And I've represented the amino acids by shapes just to show you that there's different kinds. In actual fact, there's about 20 different kinds of amino acids. And if we carry on building, we can build a protein that might look a little something like that. For some reason I've decided to go by strands and not shapes here, but that's just to show that if we magnify out, that's what it might be like. But all these different colors represent different amino acids, and those different amino acids will combine in order to make a protein with a specific shape. So there's your protein, and that can go off into the cell or wherever and go and do its job. So the overall idea is we have sections of DNA like this. This is our gene. This will code for a protein as we have just discussed, but let's go back the opposite direction. We can shrink that down, and when we shrink that down, we can compact it into our double helix. We can then zoom out a little bit again, and we'll see just the DNA looking like a piece of string. We can supercoil it in order to make it nice and compact in this chromosome, and then we can take our chromosome, shrink it back down again, shrink it back down again, and put it back in our cell. And this slide is really just to give you an idea of the level of complexity of this whole process and what level of size of molecule we're talking about. Okay, but hopefully that's made some sort of sense. Do give some feedback on this video if you feel like it, just to let me know how it went. And also it might be worth reminding you there's a list of all the videos that are available in order at the website www.mygcsescience.com. But other than that, thanks for watching, and I'll see you again soon.